genetic manipulation. It sounds like science fiction, but it's real. We are living in an age where the boundaries of what we thought was possible are being pushed every day. We can now edit the code of life. This means we can alter the very building blocks that make up every living organism. This is a huge deal. It could change everything. From the way we treat diseases to how we grow our food, the possibilities are endless. For centuries, we've bred plants and animals. This traditional method of selective breeding has been the cornerstone of agriculture. We selected for desirable traits. However, this method was far from perfect. But this process was slow and unpredictable. It often took generations to see the desired results. Now we have the power to make precise changes to DNA. This is gene editing. Gene editing is revolutionary. It allows us to make changes at the molecular level with unprecedented accuracy. It's like having a tiny pair of scissors. These molecular scissors can cut DNA at specific locations. We can use these scissors to cut and paste DNA. This means we can remove faulty genes and replace them with healthy ones. This allows us to correct genetic defects. Imagine a world where genetic disorders are a thing of the past. We can also introduce beneficial traits. The implications are huge. Imagine curing genetic diseases. Imagine crops that can withstand harsh climates and resist pests. Imagine ending hunger with disease-resistant crops. The potential to improve human health and agriculture is immense. This is the promise of gene editing, a future where we can solve some of the world's most pressing problems with the power of science. One of the most powerful gene editing tools is CRISPR-Cas9. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. That's a mouthful. But the concept is simple. Think of CRISPR as a bacterial immune system. Bacteria use CRISPR to fight off viruses. They store pieces of viral DNA in their own genome. These stored pieces act like wanted posters.
When a virus attacks, the bacteria can recognize it. They use Cas9, a special protein, to cut the viral DNA. This disables the virus and protects the bacteria. Scientists have harnessed this system. We can now use CRISPR-Cas9 to edit genes in any organism. We simply need to program the system with the right wanted poster.
CRISPR-Cas9 is incredibly versatile, but scientists are always looking for ways to improve it. They've discovered new CAs proteins with different capabilities. Some CAs proteins are better at cutting DNA in specific locations. Others can target RNA, a molecule similar to DNA. This opens up even more possibilities for gene editing.
For example, we can now temporarily turn genes on or off. This is incredibly useful for research. It allows us to study the function of individual genes. The CRISPR toolbox is constantly expanding. With each new discovery, we gain a deeper understanding of genetics, and we get closer to unlocking the full potential of gene editing.
Imagine a world without cystic fibrosis or sickle cell anemia or Huntington's disease. This is the dream of CRISPR medicine. Inherited diseases are caused by mutations in our genes. These mutations are passed down from parents to their children. They can cause a wide range of health problems. CRISPR offers a way to correct these mutations.
In the future, we may be able to cure inherited diseases before a child is even born. This is a radical idea, but it's within our reach. Clinical trials are already underway. Scientists are testing CRISPR therapies for a variety of diseases. The early results are promising. CRISPR could revolutionize the way we treat genetic disorders. Section 5. CRISPR and Cancer, a new frontier in treatment. Cancer is a complex disease. It's caused by mutations in our cells. These mutations cause cells to grow and divide uncontrollably.
Traditional cancer treatments like chemotherapy and radiation can have serious side effects. They often harm healthy cells along with cancer cells.
CRISPR offers a more targeted approach. We can use CRISPR to edit the genes of immune cells. This makes them more effective at finding and destroying cancer cells.
This approach is called CAR T-cell therapy. It's already showing promise in clinical trials. CRISPR could lead to new and more effective cancer treatments.
Section 6 CRISPR in Agriculture Sustainable Solutions for a Growing World. As we look towards the future, it's clear that innovative technologies will play a crucial role in addressing the challenges we face. The world's population is growing at an unprecedented rate, with more people living in urban areas than ever before. We need to produce more food to meet the demands of this increasing population, and we need to do it efficiently and sustainably. But climate change is making this more difficult. Extreme weather events are becoming more frequent and severe, impacting our ability to grow crops. Droughts, floods, and pests are threatening our food supply. These challenges are not just local but global, affecting farmers everywhere.
CRISPR can help us develop crops that are more resilient to these challenges. By editing specific genes, we can create plants that can withstand harsh conditions. We can use CRISPR to edit genes that control drought tolerance, disease resistance, and yield. This means crops can survive longer periods without water, resist diseases, and produce more food. For example, scientists have used CRISPR to develop mushrooms that don't brown as quickly. This reduces food waste and ensures that produce stays fresh longer. They've also developed tomatoes that are more resistant to blight, a devastating fungal disease that can wipe out entire crops. This innovation helps secure our food supply.
CRISPR could help us feed the world in a sustainable way. By making crops more resilient and productive, we can ensure that everyone has access to nutritious food. It's a powerful tool for addressing global food security. With continued research and development, CRISPR has the potential to revolutionize agriculture and help us build a more sustainable future.
Section 7. Ethical Considerations. Navigating the CRISPR Revolution. As we delve deeper into the world of gene editing, we must tread carefully. CRISPR is a powerful technology. It has the potential to revolutionize medicine, agriculture, and many other fields. With great power comes great responsibility. The decisions we make today will shape the future of humanity. We need to carefully consider the ethical implications of gene editing. It's not just about what we can do, but what we should do. One concern is the potential for unintended consequences. Even small changes in the genome can have far-reaching effects. What if we accidentally edit the wrong gene? The ramifications could be disastrous, affecting not just individuals but entire populations. What if our edits have unforeseen side effects? These side effects could manifest in ways we can't currently predict, leading to new health issues.
Another concern is the possibility of genetic enhancement. This opens up a Pandora's box of ethical questions. Should we use CRISPR to enhance human capabilities? If so, to what extent? Enhancements could range from increased intelligence to physical abilities. What traits should be considered desirable? Who gets to decide? These are complex questions that require input from diverse perspectives. There are no easy answers. The path forward is fraught with ethical dilemmas and uncertainties.
We need to have open and honest conversations about the ethics of gene editing. These discussions should include scientists, ethicists, policymakers, and the public. We need to establish guidelines and regulations to ensure that CRISPR is used responsibly. Only through careful consideration and collaboration can we navigate the CRISPR revolution safely and ethically.
Section 8. Challenges and Limitations. CRISPR's Unfinished Symphony. CRISPR is a revolutionary technology, but it's not perfect. There are still challenges and limitations that we need to overcome. One challenge is delivery. Getting CRISPR to the right cells in the body is difficult. We need to develop more efficient and targeted delivery systems.
Another challenge is off-target effects. Sometimes CRISPR can edit genes that we didn't intend to target. This can have unpredictable consequences.
Despite these challenges, the future of CRISPR is bright. We are constantly making new discoveries and developing new techniques. CRISPR has the potential to transform medicine, agriculture, and our understanding of life itself.
Section 9. The Future of CRISPR. A universe of possibilities. CRISPR is still a relatively new technology, but it's already clear that it has the potential to change the world. The possibilities are endless. In the future, we may be able to use CRISPR to develop new antibiotics to combat antibiotic-resistant bacteria, create new biofuels and other sustainable products, engineer mosquitoes that can't transmit malaria, and even resurrect extinct species, like the woolly mammoth.
These may sound like science fiction, but they are all within the realm of possibility. CRISPR is a powerful tool. It's up to us to use it wisely.